Greetings all. So today we will be presenting relating to monocrotaline present in Crotalaria genus. So for the introduction, Crotalaria is a genus of Fabaceae family which contains over 600 species. Only a few species of this genus found in temperate zones, with the majority of its representatives found in tropical and subtropical areas. Many Crotalaria species have been found to possess toxic pyrrolidazine alkaloids (PAs), including monocrotaline, throughout the phytochemical research. So the phytochemical research involving the isolation, characterization, and also structural analysis. Monocrotaline was first isolated from Crotalia spectabilis in early 20th century and exhibit biological effects such as cytotoxicity, tumorigenicity, hepatotoxicity, and neurotoxicity in both people and animals. Monocrotaline is important for agriculture and ecologies because of its strong allelophatic effects. It acts as chemical barrier, preventing generalist herbivores and altering insect behavior, while it also engages in soil-mediated interactions via root exudates and also decompose the biomass. Thus, monocrotaline signifies the complex function of secondary metabolites in the phytochemistry and ecological interactions of Crotalaria species. So monocrotaline is one of the pyrrolizidine alkaloid PA that contain high toxicity such as hepatotoxics, pneumotoxics, and genotoxics. Hence, it is not used directly in human therapy and not appropriate for direct clinical application, but it is important for indirect use in human-related biomedical research. So the toxic mechanisms can be occur where the cytochrome P450 enzyme system converts it into highly reactive and toxic metabolites that bind to the cellular macromolecules such as DNA and proteins. So how the exposure happens? The unintentional human exposure of monocrotaline can be occur when the people utilize the conventional medicinal products or consume the food items that contaminated with plant materials that contain pyrrolizidine alkaloids. So these exposures can cause the serious effects on health, including hepatic veno-occlusive disease, which is VOD, pulmonary hypertension, and also in extreme cases, it can cause death. So despite its toxicity, monocrotaline is used extensively in biomedical research, particularly in the development of experimental animal models. It is used to generate pulmonary arterial hypertension (PAH) in laboratory animals, specifically rats, to stimulate the human disease to explore process in treatments. Moreover, it is also useful in toxicology for determining the risks associated with plant-based poisons. It helps examine the neurotoxicity, genotoxicity, and also glutathione depletion. It also contributes to research on alkaloid metabolism and detoxification in the brain, particularly astrocyte-neuron interactions and also oxidative stress. So the amounts of pyrrolizidine alkaloids such as monocrotaline in food, animal products, and herbal preparations are being monitored and limited since regulatory agencies are aware of the possible harm they may cause. Despite the risk, its control application in research provides important information on toxicity and disease.
Everyone, my name is Hanani Zubair Sunar with matrix number BS2211033. So today I will briefly explain the chemical synthesis of monocrotaline, a toxic pyrrolidazine alkaloid found in Crotalaria genus. So monocrotaline or MCT belongs to a group of natural compounds known as pyrrolidazine alkaloids or PAS. These alkaloids are found in various plant families and are known for their chemical defense role in nature. Most PAS, including monocrotalines, are made up of two parts: a nisin base, which is pyrrolidazine amino alcohol, and a nicic acid, which is typically a branch mono or a dicarboxylic acid with 4 to 6 carbon atoms. When these two components combine, they form monoesters, non-macrocyclic diester, or microcyclic diesters like monocrotalines. From a synthetic chemistry perspective, these structures poses a challenge. The bicyclic natures of the nisin base and the flexibility of the ring system, combined with the few stereocenters, make total synthesis difficult, but also appealing for organic chemists. Several synthetic pathways have been developed to mimic the natural formations of these compounds, particularly to replicate the ester linkage that bind the niacin and the nitric acid units. Interestingly, monocrotaline is not used therapeutically due to its high toxicity. Once inside the body, especially in astrocytes, it is activated by the cytochrome P450 enzyme system, producing toxic intermediates like DHP and DHMC. These compounds can modify cell proteins and damage organs such as the liver, lungs, and brain. However, despite its toxicity, monocrotaline has proven extremely useful in medical research, especially for creating animal models of pulmonary arterial hypertension or PAH. By studying how monocrotaline is induced PAH, researchers are able to explore molecular pathways involved in the disease, which contribute to new insights in cardiovasculars. Crotalaria species also naturally produce as a defense toxins against herbivores used by insect in pheromones and predators avoidance systems. In conclusions, monocrotaline serves as both a toxic natural compound and a valuable research tool. Its synthesis highlights the complexity and potentially of natural product chemistry, especially when derived from plants like Crotalaria retusa. Thank you.
Pyrrolizidine alkaloids or PAs are toxic secondary metabolites produced by certain plants to protect themselves from herbivores. These compounds have characteristic bicyclic structure and vary widely depending on the plant species. Today I will briefly explain how these molecules are biosynthesized in Crotalaria sp. The first step is amino acid precursors. The biosynthesis starts with the amino acids L-ornithine or L-arginine, which are converted into putrescine, a simple diamine involved in many metabolic pathways. This putrescine undergoes oxidative deamination by the enzyme diamine oxidase, producing 4-aminobutanol. This intermediate then spontaneously cyclized to form pyrrolinium cation, the key precursor to the niacin base. The second step is the niacin base formation. Two molecules of the pyrrolinium cation condense to create the pyrrolizidine skeleton, known as the niacin base, such as retronicine. This step is highly specific and likely catalyzed by an enzyme. The niacin base forms the core bicyclic ring structure found in all pyrrolizidine alkaloids. The third step is the nitric acid and esterification. Meanwhile, the plant produces nitric acid through branched-chain fatty acid synthesis. These acids usually contain 4 to 6 carbons, may be further modified by hydroxylation or methylation. The niacin base then reacts with one or more nitric acids in an esterification reaction catalyzed by acetyltransferase enzymes. This results in different PA types: monoesters, non-macrocyclic diesters, and macrocyclic diesters. For example, the monocrotaline, a toxic monocrocyclic diester, which form this way. The fourth step, storage and defense. Once synthesized, the PAs are stored in the plant as N-oxides, which are more water soluble
Hello everyone, my name is Fatin Hanizati binti Muhammad with matrix number BS22110394. Today I will be talking about biological activity of monocrotaline. So, the first one is anti-cancer effect. Monocrotaline has shown cytotoxic effects against HepG2 liver cancer cells in lab studies. That means it can kill or inhibit these cancer cells. It has an IC50 value of around 25 micrograms per milliliter, which indicates how potent it is. The lower the number, the stronger the effect. The response was dose dependent, so the more monocrotaline used, the greater the cancer killing effect. On a molecular level, it interacts with important cancer-related proteins like p53, HGF, and TREM1, which are all involved in tumor growth and survival. Interestingly, monocrotaline also showed non-mutagenic effects, and it had low toxicity to normal cells like human fibroblasts. Besides its anti-cancer effect, monocrotaline also has anti-parasitic activity, especially against a parasite called Trichomonas vaginalis, which causes a common STI. In lab experiments, monocrotaline inhibited about 80% of the parasite's growth at 1 mg per milliliter. Even better, it showed no harmful effects on healthy human epithelial cells, meaning it was effective but safe, at least in those early tests. Also, other pyrrolizidine alkaloids like useramine have been shown to reduce biofilm formation by bacteria like Staphylococcus epidermidis. So, to sum it up, monocrotaline shows strong anti-cancer potential, especially for liver cancer, and it also has promising anti-parasitic and anti-biofilm effects. Even though it's a toxic compound in high doses, these biological activities show that with proper modification, monocrotaline could be developed into new treatments for diseases that are hard to fight with current drugs. Thanks for listening.
To conclude, production of monocrotaline significant in crotalaria through complex biosynthesis. It may be toxic but also shows real scientific value. It has promising biological activity, especially in fighting cancer and parasites, while staying less harmful to healthy cells. Monocrotaline is a compound of scientific interest not just for its risk but for its potential. The biological effects, natural biosynthesis, and chemical challenges continue to drive research in pharmacology and toxicology.